welcome back to Tales of the Resistance. I'm Mara. I'm one of the regular hosts and a member of the I Am Responsible team here, as always, with Beth, my usual co-host, and also on the I Am Responsible team with me. Say hello, Beth.
Hello, everybody.
And so this podcast is all about antimicrobial resistance. We've been on a little bit of a hiatus, but we're coming back with some interviews with folks we've been working with who are doing research on antimicrobial resistance from an environmental perspective. They're mostly working with water quality monitoring for antimicrobial resistance. And so we're going to explore some of the different ways that they are measuring for antimicrobial resistance in an environmental setting, and also how they're putting all that information together to try to understand what it means for human health. So we're going to have several of these interviews here, and hopefully you guys will find it as interesting as we did. We certainly learned a lot from these discussions. So in this episode, we got to sit down with Michelle Soupir. She is the PI of this project, which is
what if someone doesn't know what a PI is?
I'm going to get to it.
Okay.
You're introducing me. I mean, you're interrupting me. This is a multi-university project between the University of Buffalo, the University of Iowa State, and of Nebraska. And as the PI, that means she's the principal investigator and she's the leader of the project. So we got to get in with Michelle to talk a lot about what the background was of how they got started on this, and also some of the things that they're trying to learn. It was a very interesting conversation.
Yeah, today was really interesting. I learned a lot. You can really tell that Michelle has 17 years of experience. She has a really in-depth knowledge.
All right, great. Well, I'm going to go ahead and transition us over there, and we'll talk to you again soon. All right, welcome. Michelle. It's great to have you on the podcast. We have got to talk before on different parts of this project that we're working on here together, but it's great to have you on the podcast, and just give everybody a little bit of a background about who you are and what you're about.
Okay, so I am Michelle Soupir. I'm a professor in agricultural and biosystems engineering at Iowa State University, and I'm currently serving as the principal investigator on this project that's evaluating the essentially the risk to human health from agricultural sources of antimicrobial resistance. So a little bit about my background. I grew up in Kansas and went to undergraduate at Kansas State University. There I've kind of been in ag and biosystems or agriculture and biological systems engineer throughout my entire career. It just happens to be named different departments at different universities. So, but really wanted to work in the environmental science field, and then also to, take kind of that science and apply it to engineering designs. So, with the kind of overall goal of being able to understand, what's happening out in the environment, and then design solutions to try and mediate that impact. So I actually after I graduated from Kansas State, I worked at an environmental engineering consulting firm in Kansas City, and worked on kind of remediation projects, kind of the HAZWOPER projects that were super fun projects through the U.S. Environmental Protection Agency. Specifically, I was working in downtown Wichita on a groundwater remediation project, and that was really interesting to kind of see how we were mediating contaminated sites. But as I was thinking about going back to school for a master's degree, I wanted to be, if I could, a little bit more on the prevention side. So instead of dealing with a problem that's been, happening out in the environment for a really long time. Now that we know so much more about what we're releasing into the environment, could we work to prevent things from actually moving off-site and getting into water systems and then being exposed to humans through those processes? And so I went to Virginia Tech for my graduate degrees, master's and PhD, and there is where I was really able to kind of move into kind of the animal manure management space. So, animal production, of course, really important. People are going to eat meat. And so from that, we have this product, manure, which can be very beneficial to agricultural lands if it's applied and managed at agronomic rates and managed in a way that it's not spilling out into the environment or over applied. And so what I really had the chance to do at Virginia Tech, though, then was to start working on the microorganisms that are present in that manure. So the manure is beneficial because of the nutrients to help enhance crop production. Micronutrients are present also, but because it's from a living organism, we also have the potential for pathogens and also a lot of commensal microbial organisms that are located and present in that manure, surviving in that manure. When it's spread out into agricultural fields, we want to be able to mitigate it so that it doesn't move into water systems and pose a risk to human health. And so there's some different strategies that we can look at. A lot of my work has been, after the manure has been applied to the land, thinking about how can we better prevent that movement, whether it's through different types of equipment used to apply the manure to the fields, looking at the timing of the manure. So manure can often be applied kind of in the late fall timeline or in the early spring before planting, and that can have different impacts on the movement of E. coli or other pathogenic organisms. And then we've also done some work looking at on-farm, so how to prevent the pathogenic organisms from, doing some kind of treatment before that manure is applied to the land. So after as I completed my PhD at Virginia Tech, then I came to Iowa State and began my career here. And so it's been, I've been at Iowa State 17 years, and over that timeline, it's been really interesting to see kind of what are the contaminants of concern. We still talk a lot about nutrients in the state of Iowa, looking at the movement, especially of nitrogen, because we have soils that need to be drained in order to be productive for agriculture production. And so because of that, the drain lines are installed underneath the agricultural fields, and this moves the water off-site much more quickly than our natural prairie and wetland systems. And so we've been designing a lot of different conservation practices that can be installed kind of at the discharge point. So where that water moves off the fields, can we do some kind of edge of field type of treatment to prevent that nitrogen from moving directly into the waterways? So, and that's a microbial process, microbial denitrification. We're trying to create systems that can kind of create those ideal environments for that community of microorganisms that work together to try and remove that nitrate and convert it to nitrogen gas before it moves into the water system. And then when I really came to Iowa State, had much more experience working with E. coli. So E. coli is an organism that's used to track the potential. It's a fecal indicator organism, so it's used to track potential pathogens in the environment. The reason we work with E. coli is it's safe. Most versions of E. coli are going to be harmless. There are, of course, the strains that can cause a lot of problems or make a lot of problems for human health, problems for humans. But for the most part, a lot of E. coli is commensal. It's present in the GI tracts of humans, animals. And so from there, it's something that can be an indicator. If E. coli is present, then there could be other microorganisms that could have more negative human health impacts. And so we have criteria that have been set by the Environmental Protection Agency, and we monitor waters throughout the state, throughout the United States, to see what those levels of E. coli are, and then we can use that to classify a water body as far as if it's safe for usually recreational purposes. And so then as I was here, excuse me, at Iowa State, after a couple of years, the antimicrobial resistance became I would say it was a very much acknowledged in the human health space. We were already seeing mitigation as far as reducing prescription usage and making sure that a antibiotic was necessary in order to treat an infection. Not quite the wide spread distribution of antibiotics that we would see when I was younger and my parents were younger, but much more focus on conserving the use of antibiotics. But then, of course, in agriculture, we do use antibiotics for at this point, primarily disease prevention or for treatment of sick animals. Some antibiotics can be used for growth promotion, although that would be antibiotics that are not classified by the Food and Drug Administration as being, a certain level of importance to human health. And so with that, we began to look at the presence of antimicrobial resistance in some of the animal management systems that we have here in Iowa. First, we focused on swine, and that's really been kind of our focus even now because of the large amount of swine production in the state of Iowa.
Mm-hmm. Can I ask, I know you talked a little bit about how your research got into AMR, but we always ask everybody is how they first got introduced to antimicrobial resistance. Where did you first hear about it? First understand that it was an issue, a meaningful issue to you in some way.
Yeah. So I don't know that I have a real personal story with AMR. I can tell you that my colleague at the time, Alok Bhandari, we had been monitoring the water quality at one of our research farms for Iowa. It's near Nashua, Iowa, so it's in the northeast, about 2 hours from the Iowa State University campus. And that site is really unique because it has different treatments of different types of management and then also manure application. So it's one of our farms that we can really focus and inform in this case, the swine industry about best practices for manure management. And so we really had an opportunity up there because we had, a producer that would allow us to get the manure samples. And then we could start to look and see what kind of resistance was present in that manure. So that was, really the first time that I started thinking about manure in agriculture. I actually was, I would say, very unaware that antibiotics were necessary for animal production. I think because I grew up, my grandpa did farm, but it would be, like 20 to 50 cattle, and they were mostly pastured, so it wasn't the intensive kind of agriculture that we've really seen develop, especially probably the last 30 years. And so I really didn't have awareness, just coming from more of a beef system, of the kind of intensity of raising animals, and that with so many animals so close together that if someone gets sick, it can spread very quickly, and hundreds of animals or maybe even thousands of animals can become ill. And so I guess I, was kind of naive as far as recognizing the impact of and the scale of antibiotic use in agriculture.
Can I take you back a little bit? Because you got to talk about your career and how you got into this area, but maybe you could introduce this specific project a little bit more in terms of why you chose it. What are the kind of different areas of exploration that you guys are doing and so forth? What are the goals and things like that?
Yeah, you bet. So our first project was looking at kind of the movement of some resistant phenotypes of E. coli and Enterococcus. Enterococcus is another indicator organism. from swine systems that had been applied to tile drained ag land, and then seeing if those phenotypes could move into the drain lines, and then if we could detect them, kind of at the edge of the field. And so we did a lot of work looking at kind of understanding the movement of these resistant organisms from manures that had been applied to usually corn and soybean systems, sometimes continuous corn. And then we've really been able to build on that over the years, and especially in partnership with Dr. Adina House, it's really been, she was hired in 2015, and when she was hired, she brought in the ability to do a lot more work with the genetics. So we were just looking at using kind of PCR to kind of determine if there were different genes of resistance that were present in the organisms that we plated. And then when she joined Iowa State, she was able to really expand the way we could explore the resistance and how it was defined within a group of microbial organisms or a microbial community. So now, with the equipment that she has and her expertise in data analysis, we can tell a lot more about a much wider set of resistant genes. We know what those genes can indicate. Maybe they are telling us that there's resistance to a certain class of antibiotic. Maybe it's telling us that there's resistance or that the genes are on plasmids and they can move from organism to organism. She's just really been able to help us expand our understanding of that resistance that's in the environment. And so we've had a couple of additional projects since then. One of my really favorite projects I've ever had the chance to work on, we looked at prairie strips. So prairie strips can be integrated into agricultural lands. Essentially, they're in strips, and then we know they have a lot of water quality benefits in that they can, filter out sediment, they can increase infiltration, but we wanted to see if those prairie strips could be effective in reducing any antimicrobial resistance that was coming into the prairie. And when we got that, we also partnered with Diana Aga at the University of Buffalo for the first time. And she's really a renowned chemist and was able to look at the not only the antibiotics, but their byproducts, the metabolites of the parent compound, so that we could also start to understand how the antibiotics are breaking down and behaving in the environment. And because there's some concern that, if the parent compound isn't a risk, maybe one of the metabolites could actually be more harmful. And so we're able to start looking at those different, metabolites of the parent compounds. We had information on the antibiotic use sometimes, and so we could kind of start to tie all of those pieces together. So it was a really unique team because we had our expertise in environmental fate and transport monitoring. So we brought in Dr. Howe and her team and understanding that microbial community. And then we brought in Dr. Aga and her team and understanding kind of the chemical composition. And so we had a couple of projects with them. And then most recently is when we started to collaborate with the team from University of Nebraska-Lincoln. So Dr. Bartlett Hunt had a watershed that she has done a lot of work in, and so we're able to kind of expand the geographic location. I've mentioned tile drainage. We have soils that hold on to the water, and so we actually have to drain the soils in many parts of Iowa. Very different hydrology in Nebraska, sandy soils, irrigation, and so that gave us the chance to really kind of expand what we've done in Iowa to a different state, a different ecosystem, and then also to build on and collaborate with the good work that had already been done in Nebraska. And then we brought in Dr. Amy Schmidt, because she has this broad reach through social media and other outlets, so the I Am Responsible platform. And then Dr. Bing Wang does the risk assessment. So we needed someone who could connect what was happening, like what we're detecting in the environment. We have really unique information about what these samples contain, but what is that actual risk to human health? And so she's the person that can make that connection. She does a lot of risk assessment from the environment, also from foods, to try and help us understand, like, if we just say there's all this stuff in our water samples, someone can say, so what? Like, what does that mean if we can't really connect it to, like, does it actually impact humans when they're exposed to this? How much do they need to be exposed to? Which of these, 100 different resistant genes and compounds actually have an impact? And so she's the one who's really been able to help us start moving in that risk assessment direction. And I also I should have mentioned Dr. Dan Anderson is our other collaborator here at Iowa State. And he's the person who does all the manure management. So he works very closely with folks all over the country and does a lot of manure management outreach and education. He does the manure applicator training for the state of Iowa. And so that's been a really great way for us to kind of disseminate the results of our studies as we have them, and try to get manure applicators to, implement some changes in how they do their management and manure application based on the research that we're doing at the three institutions.
Well, I was going to actually kind of this, and then we were mentioning earlier where you're talking about in the manure application training, what recommendations are coming out of this research in terms of how people should apply manure, the prairie strip usage. So I'm just kind of curious about what are the results showing in terms of practices that people could implement either in manure application or like the prairie strip more generally in crop production.
Yeah, the manure application has been really an interesting piece, because we did see that fall manure application allowed for die-off of the microorganisms over the winter. And so because of that, there was less risk of movement off-site, especially drainage into the tile lines if the manure was applied in the fall. Of course, from the nutrient perspective, we don't want to apply manure too early. We want it to be applied late enough in the fall that we don't, lose those nutrients or have a chance for them to run off. So there's kind of it's a little bit tricky, but later in the winter was better for that die-off to occur, because if the manure application took place in the spring, then those organisms were, still alive, and then temperatures were becoming warmer, which were kind of ideal conditions for survival. And so then they were more likely to move off-site. There is, some nutrient and water quality benefits from that spring manure application, just because it's applying closer to the time when the crops will need to use those nutrients. And so there's less of a time period of off-site movement. Another thing, we're encouraging cover crops. So if you apply manure in the fall, if you can get a cover crop on, that can also help to stabilize the nitrogen and reduce the nitrogen loss. And then that overwinter kill of the microorganisms can help to keep the pathogenic organisms off the field. So that's been one thing. I know that we did see some buildup of resistance in the soil in the continuous corn. But when we had a kind of a corn and soybean rotation, you would only typically apply that manure prior to planting corn. You wouldn't typically apply manure prior to planting soybeans, because they are able to fix nitrogen from the environment. And so because of that, by providing that, every other year application timeline instead of every single year, that seemed to allow for more time for die-off and mitigation to occur relative to kind of the continuous corn. Dr. Howes led a study looking at different types of treatment to try and reduce the antibiotic resistance in the manure, and it found that anaerobic digestion, I think, performed the best. I know there's a paper that we just got published on that I could share. And that is, again, was unique because we were able to assess both the phenotypic resistance, the genotypic resistance, as well as the antibiotics. But of course, if one practice was really good at the killing off the live organisms, then something else would, then you might see an increase in the resistant genes. So
Yeah.
it's messy. I wish it was always a cleaner picture, but anaerobic digestion did seem to have kind of the most impact for overall mitigation prior to land application. And then our study with the prairie strips really showed a good reduction in, I think it was definitely the resistant genes and the phenotypic organisms in the surface runoff. And so that was a really promising strategy. One other thing that I guess back to the project that we have, the monitoring in Iowa is focusing on the Black Hawk Lake watershed. And that is an area where we have small subwatersheds with different levels of conservation practice implementation. So one watershed was at the beginning of the study, there were very few conservation practices, and then a similar watershed right next to it had high levels of conservation practices. So, and over time that has changed, but at one site we now have a wetland, and then the other site there is, some practices, but some practices we've also seen some disadoption. So there was a couple of years of cover crops, and then those cover crops were not planted more recently. So we saw a movement to try out a conservation practice and then back off of it. But then we've also seen the implementation of some more structural practices, such as wetlands, which then are more permanent. And so that's been really nice to be able to compare the impacts of both the conservation practices as well as the manure, and try to kind of understand the more complicated impacts of watershed scale monitoring and different inputs from a lot of different sources, rather than just kind of these individual studies that we first started doing that were, really informing individual practices. And now this study is looking at more of those combined impacts.
Mm-hmm. Interesting. I know you mentioned both cover crops and prairie strips. In Nebraska, we have a lot of grass waterways. Not a lot of, that's a lot of the most popular way of having a buffer strip. Would a grass waterway, without all that diversity, would it be enough, really, to make an impact?
Yeah, I think that grass waterways have a lot of impact too. They may not have quite as deep of a rooting system, so those prairie strips can be like 20 feet. The roots in the prairies can be like 20 feet deep, which because of that you get the development of macropores is what they're called, where little, like earthworms are like creating these direct pathways which allow that water to infiltrate very deep, whereas a more of a shallow stand of grass, it depends on what kind of vegetation is selected for those, but that may not allow for quite as much infiltration as the prairie strips do. But then from the surface processes, it should be very similar, right? Because you've essentially got a filter that's physically straining that water and allowing, slowing the water down, which then allows kind of sediment to settle out. Also, as the water passes through that grass, bacteria can stick to the grass and kind of serve as an actual, living filter to try and reduce some of those pollutants from moving through the system. So yeah, in a lot of ways they're, very similar.
Yeah. Okay.
I kind of want to go back to as you're talking to folks about the results you're getting. Are you getting a lot of interest? Does it seem like people are excited about implementing some of these, and are they being at all like that decision making driven by antimicrobial resistance, or would you say it's more other things that they're being engaged on?
Yeah, I think it's tricky because we do not have we don't have a lot of monitoring for antimicrobial resistance, and so in general, just it's still kind of novel to know like what resistance is out in the environment. We don't, and different groups and different labs are looking at different indicators, so there hasn't been kind of this national uh, well, there has been some work by the EPA and USGS, so I don't want to say there hasn't, but there hasn't been kind of the widespread type of monitoring that we see for E. coli or some nutrients. So, kind of the more common pollutants that we've been monitoring for a really long time. We just don't have those kinds of data sets on AMR. And then the other thing that's really tricky, which is, what we hope to contribute with the study is all this stuff is out there, but what does it mean, right? And it's so expensive to monitor for hundreds of different chemicals and different antibiotic resistant genes. Which ones are really important? Which ones actually mean something? So trying to get that risk to human health. I think that most people would still say like the environmental pathways of antimicrobial resistance are still really understudied. We know that resistance can build up in populations in like the stream bed sediments. We know like downstream discharges from especially things like wastewater treatment plants. Those stream bed sediments can have high concentrations of resistance and different pollutants. But then 20 miles downstream, concentrations are much more dilute. And, so what kind of exposure is going to be a problem? What kind of education makes sense? Like, don't get into the water if you have an open wound. If it's an area, you may need to use hand sanitizer or take a shower when you get home, just so that you don't have, things that can, persist on your body. So, that it's been really tricky though to try and put it's very much still in a new developmental state as far as what should we even be monitoring for, and then what does that mean. So and I hope that we can get to that with this project, but also there's been so much method development, developing which genes should we be monitoring for in the environment, which chemicals are important to monitor in the environment. We want to really for a risk assessment, we need to be able to predict the movement of these different organisms and chemicals in the environment. And so we're using a model called the Swollen Water Assessment Tool, but that model has to be modified because it's good at predicting hydrology and nutrients, but it needed to be expanded with another component for resistance. So there's been a ton of work in that space. And then even the methods for assessing risk from environmental exposures. I know Dr. Wang has really been working with her student to advance that. So there's just a ton of little pieces along the way that needed a lot of method development in order to kind of help tie it all together. So that has been tricky in the communication piece, because I don't think we're to a point where we have a real solid story. I can't say that if there's a swine farm upstream and you go canoeing, you're going to get sick, right? We can't say things like that because we don't have kind of those hard connections to be able to really explain the impact of the things that we've been able to detect in the environment.
Mm-hmm. Mm-hmm. All right, so we'll probably need to leave it there for today, but thank you so much for taking the time, and we got to learn quite a bit about obviously the project, but also all the different things went together today. So that was really good.
Yes,
Yeah.
thank you so much. I appreciate meeting you and everything.
Yeah, you too.